going to present how DNA is arranged in the cell. So, as you remember from my previous class, I have presented the DNA, RNA, protein, DNA replication, transcription, translation, and many more things. Now, the topic is how DNA is arranged in the cell. So, DNA in the cell is arranged as a double helix, similar to a spiral staircase with its two strands forming the structure. In eukaryotic cell, this DNA is then bound tightly around histone proteins to form nucleosomes. These nucleosomes are further coiled into chromatin, which condenses into chromosomes. The large visible structure that house the DNA within the cell's nucleus. In contrast, prokaryotic cells lack a nucleus and contain their DNA as a coiled structure within the cytoplasm called a nucleoid. Now, the in general, the size of the genome is in one of the most well-studied prokaryotes, that is E. coli, is 4.6 million base pairs. So, which would extend a distance about 1.6 millimeter if it stretched out. So, how does this fit into a small bacterial cell? The DNA is twisted beyond the double helix in what is known as supercoiling. Some proteins are known to be involved in supercoiling. Other proteins and enzymes help in maintaining the supercoiled structure. Now, the double helix structure contains nucleotides, complementary base pairing, the helix. Nucleotide DNA is made up of building blocks called nucleotides, each consisting of a sugar phosphate group and a nitrogenous base, that is ATGC. Now you can see ATGC, adenine, thymine, guanine, cytosine. Now, complementary base pair, these nucleotides form two long strands with the bases pairing up in a specific way. For example, adenine always pairs with thymine by using two hydrogen bonds. Then cytosine always pairs with guanine by three hydrogen bonds. As you can see, these base pairs are here. Now, the helix, the two strands twist around each other with the sugar phosphate backbones on the other outside and the base pairs in the interior forming a double helix. Now, so you can see the base pair, sugar and phosphate backbone, then major groove and the minor grooves 5 prime to 3 prime, 3 prime to 5 prime. Now, the next is the packaging of DNA. Histones and nucleosomes. In eukaryotes, negatively charged DNA wraps around positively charged histone proteins to form a structure called nucleosomes. Chromatin, these nucleosomes are then coiled and compacted into a material called chromatin, which fills the nucleus. Chromosomes, during the cell division, chromatin further condenses into the distinct thread-like structures known as chromosomes. Now, cell types, eukaryotes and prokaryotes. Like eukaryotes, like those of plants and animals, have their chromosomes organized within a membrane-bound nucleus, while prokaryotes, like in bacteria, do not have a nucleus and their DNA is located as a single circular chromosome within the cytoplasm in a region called the nucleoid. Now, the organization of eukaryotic chromosomes, double helix DNA, you can see here, DNA wrapped around histone, which you can see here. Now, nucleosomes coiled into a chromatin fiber, which you can clearly see here. Further condensation of chromatin, which is clearly available now. Duplicated chromosomes is available here. So, if you will take the minor part of chromosome, you will observe this, and if you take further the minor part, then you get this visibility, and then this, and then this. Now, if you want to understand the final packaging of DNA, then this video is worth for you to understand how in exactly this DNA to protein process happens. So
Here is a
please have a
cell,
look.
the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Now, after seeing this, we can move to the next part, that is differences between prokaryotic and eukaryotic replications, which we have discussed earlier also, but still we can see origin of replication single in prokaryote, while in eukaryotes it is multiple. Rate of replication 1000 nucleotides per second, while in case of eukaryotes 50 to 100 nucleotides per second. Chromosome structures are circular in prokaryotes while linear in eukaryotes. Telomere is not present in prokaryotes while it is present in eukaryotes. Now, the DNA repair, which is most significant part, playing roles in major human diseases, especially the cancer. So here you can see the DNA damage and repair process. So DNA polymerase can make mistakes while adding nucleotides. It edits the DNA by proofreading every newly added base. Incorrect bases are removed and replaced by the correct base, and then polymerization continues. Most mistakes are corrected during replication, although when this does not happen, the mismatch repair mechanism is employed. Mismatch repair enzymes recognize the wrongly incorporated base and excise it from the DNA, replacing it with the correct base. In yet another type of repair, nucleotide excision repair, the DNA double strand is owned and separated. The correct incorrect bases are removed along with the few bases on the 5' to 3' end, and these are replaced by copying the template with the help of DNA polymerase. Nucleotide excision repair is particularly important in correcting thymine dimers, which are primarily caused by ultraviolet light. In a thymine dimer, two thymine nucleotides adjacent to each other on one strand are covalently bonded to each other rather than the complementary bases. If the dimer is not removed and repaired, it will lead to a mutation. Individuals with flaws in their nucleotide excision repair genes show extreme sensitivity to sunlight and develop skin cancers in early life stage. Most mistakes are corrected. If they are not corrected, they may result in mutation, defined as a permanent change in the DNA sequence. Mutations in repaired genes may lead to serious consequences like the cancer. So there are different causes of DNA damage. For example, the X-rays, ultraviolet rays, X-rays, anti-tumor agents, replication errors. Now, there are different process of repair. For example, base excision repair, BER, nucleotide excision repair, NER, recombinatorial repair, that is HR or NHEJ, then mismatch repair, MMR. So for these things also we have included a video, sub-videos, which can clearly define the DNA damage and repair process. This figure has been adapted from the book, which is also sufficient for basic understanding of DNA damage and the repair process. Now, the mechanism of DNA repair we have discussed already. For example, here, single strand break, single base damage, bulky lessons, cross links, base mismatch, then double strand break. So there are well-defined pathways for each of these mechanisms, which you can also read the review articles regarding DNA damage and repair mechanisms in details. Now, this is the another video which presents the details of DNA damage and
just
repair
one of your
process.
cells gets damaged tens of thousands of times per day. Multiply that by your body's 100 trillion or so cells, and you've got a quintillion DNA errors every day. And because DNA provides the blueprint for the proteins your cells need to function, damage causes serious problems, such as cancer. The errors come in different forms. Sometimes nucleotides, DNA's building blocks, get damaged. Other times, nucleotides get matched up incorrectly, causing mutations. And nicks in one or both strands can interfere with DNA replication, or even cause sections of DNA to get mixed up. Fortunately, your cells have ways of fixing most of these problems, most of the time. These repair pathways all rely on specialized enzymes. Different ones respond to different types of things. One common error is base mismatches. Each nucleotide contains a base, and during DNA replication, the enzyme DNA polymerase is supposed to bring in the right partner to pair with every base on each template strand, adenine with thymine and guanine with cytosine. But about once every 100,000 additions, it makes a mistake. The enzyme catches most of these right away and cuts off a few nucleotides and replaces them with the correct ones. And just in case it missed a few, a second set of proteins comes behind it to check. If they find a mismatch, they cut out the incorrect nucleotide and replace it. This is called mismatch repair. Together, these two systems reduce the number of base mismatch errors to about 1 in 1 billion. But DNA can get damaged after replication too. Lots of different molecules can cause chemical changes to nucleotides. Some of these come from environmental exposure, like certain compounds in tobacco smoke. But others are molecules that are found in cells naturally, like hydrogen peroxide. Certain chemical changes are so common that they have specific enzymes assigned to reverse the damage. But the cell also has more general repair pathways. If just one base is damaged, it can usually be fixed by a process called base excision repair. One enzyme snips out the damaged base, and other enzymes come in to trim around the site and replace the nucleotides. UV light can cause damage that's a little harder to fix. Sometimes it causes two adjacent nucleotides to stick together, distorting the DNA's double helix shape. Damage like this requires a more complex process called nucleotide excision repair. A team of proteins removes a long strand of 24 or so nucleotides and replaces them with fresh ones. Very high frequency radiation, like gamma rays and X-rays, cause a different kind of damage. They can actually sever one or both strands of the DNA backbone. Double strand breaks are the most dangerous. Even one can cause cell death. The two most common pathways for repairing double strand breaks are called homologous recombination and non-homologous end joining. Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes interlace the damaged and undamaged strands, get them to exchange sequences of nucleotides, and finally fill in the missing gaps to end up with two complete double-stranded segments. Non-homologous end joining, on the other hand, doesn't rely on a template. Instead, a series of proteins trims off a few nucleotides and then fuses the broken ends back together. This process isn't as accurate. It can cause genes to get mixed up or moved around, but it's useful when sister DNA isn't available. Of course, changes to DNA aren't always bad. Beneficial mutations can allow a species to evolve, but most of the time, we want DNA to stay the same. Defects in DNA repair are associated with premature aging and many kinds of cancer. So if you're looking for a fountain of youth, it's already operating in your cells. The DNA.
Now, I'm showing the picture for the translation, which begins when a transfer RNA anticodon recognizes a codon on the mRNA. The large ribosomal units joins the small subunit, and a second transfer RNA is recruited. As the mRNA moves relative to the ribosomes, the polypeptide chain is formed. Entry of a release factor into a site terminates translation and the components dissociate. Now, how the genes are regulated. This is also very interesting topic, which you need to understand how in exactly the genes are regulated in case of eukaryotes and prokaryotes. For a cell to function properly, necessary proteins must be synthesized at a proper time. All the organisms and cells control to or regulate the transcription and translation of their DNA into a protein. The process of turning on a gene to produce RNA and protein is called gene expression. Whether in a simple unicellular organism or in the complex multicellular organism, each cell controls when and how its genes are expressed. For this to occur, there must be a mechanism to control when a gene is expressed to make RNA and protein, how much the protein is made, and when it is time to stop making the protein because it is no longer needed. So, cells in multicellular organisms are well defined and specialized. Cells in different tissues look very different and perform different functions. Now, this is the video which will help you to understand the
This is the lac operon.
gene
Here
expression.
you see three structural genes, LAC-Z, LAC-Y, and LAC-A. LAC-Y encodes the enzyme permease, which is necessary for allowing lactose to enter the cell. LAC-Z encodes the protein beta-galactosidase, and LAC-A encodes the protein transacetylase, both of which play a role in the catabolism of lactose once it's inside the cell. All three of these proteins work together to help the cell bring in and metabolize lactose when it's available in the environment. This breakdown of lactose provides energy for the cell. These genes all share a promoter and an operator. Further upstream of the operon, you see the regulatory gene LAC-I, which encodes an active repressor. When there's no lactose in the environment, this repressor can bind to the operator. This prevents RNA polymerase from binding to the promoter of the operon, which therefore limits the expression of the LAC genes. This is beneficial to the cell because it wouldn't want to waste the energy and resources to make the proteins to break down lactose when lactose isn't present in the cell. For these LAC operon structural genes to be expressed, two things need to happen. First, the repressor needs to be deactivated, so it cannot bind to the operator and prevent RNA polymerase from transcribing the LAC genes. For this to happen, lactose needs to be present. When lactose is present, allolactose can be derived from it. Allolactose acts as an inducer for this operon by binding to the repressor, changing the shape of its DNA binding domain, and making it inactive. In this way, the presence of lactose helps promote transcription of the genes necessary for lactose catabolism. The second thing that needs to take place for the lac operon structural genes to be transcribed is the binding of the CRP cyclic AMP complex to the promoter. Cyclic adenosine monophosphate, also called cyclic AMP, is a small nucleotide that is synthesized from ATP by the enzyme adenyl cyclase. Cyclic AMP receptor protein, which I'll call CRP, is a DNA binding protein that, when attached to cyclic AMP, will bind to the lac operon promoter. When the CRP cyclic AMP complex binds to the lac operon promoter, it can help RNA polymerase interact with the promoter and transcribe the lac operon genes. So, for transcription of the lac genes to occur, the repressor needs to be deactivated by the presence of lactose, and the CRP cyclic AMP complex needs to be available to bind to the promoter to help RNA polymerase attach to the DNA. So what controls whether or not the CRP cyclic AMP complex is available to bind to the lac promoter? That has to do with the availability of cyclic AMP in the cell. And the cyclic AMP availability is impacted by the presence of glucose in the cell. When both lactose and glucose are present in the cell's environment, glucose is the preferred molecule to break down. The cell can more efficiently use glucose than it can lactose, so it will repress the lac operon even though lactose is present. This allows the cell to focus on the use of glucose for energy. The presence of glucose in the cell decreases the activity level of the enzyme adenyl cyclase. This means that in the presence of glucose, adenyl cyclase activity is low, which means cyclic AMP levels are low. In these instances, there isn't enough cyclic AMP available to bind to CRP. Without the CRP cyclic AMP complex interacting with the lac operon promoter, RNA polymerase cannot attach as effectively to the promoter of the operon, and transcription of the lac operon structural genes cannot occur. So when glucose is available, even if lactose is present and the repressor of the operon is inactive, the lac genes are not expressed. This is known as glucose repression because the presence of the preferred catabolite, glucose, prevents the expression of the lac operon. You can see that the lac operon takes advantage of two different regulators, the lac repressor and CRP, to broaden the range of environmental cues the operon responds to. The lac repressor is considered a negative regulator because when it's active, it prevents RNA polymerase from binding to the promoter. Remember that the lac repressor responds to the environment by becoming inactive in the presence of lactose. CRP is considered a positive regulator because when bound to by cyclic AMP and attached to the promoter, it supports RNA polymerase binding to the promoter. Remember that the CRP cyclic AMP complex binds to the lac promoter in the absence of glucose. Using these two regulators together means the cell can respond appropriately to the mix of glucose and lactose in the environment, allowing the cell to be as efficient as possible, breaking down glucose instead of lactose when they're both present, but relying on lactose when glucose is not available. So to review, in the absence of both lactose and glucose, the lac-I repressor is active, so the lac operon genes are not expressed. In the presence of both glucose and lactose, the lac-I repressor is inactive, but the CRP cyclic AMP complex is not available to bind to the promoter.
Now, prokaryotic versus eukaryotic gene expression. To understand how gene expression is regulated, we must understand how a gene becomes functional. Protein and in a cell. The process occurs in both prokaryotic and eukaryotic cells, just in slightly different fashion. Because prokaryotic organisms lack a cell nucleus, the process of transcription and translation occur almost simultaneously. When the protein is no longer needed, transcription stops. As a result, the primary method to control what type and how much protein is expressed in prokaryotic cell is the regulation of DNA transcription into RNA. All the subsequent steps happen automatically. When more protein is required, more transcription occurs. Therefore, in prokaryotic cells, the control of gene expression is almost entirely at the transcriptional level. The first example of such control was discovered using E. coli in 1950 and 1960, that is called operon, and for which I have shown the videos. Now, the different process, you can see the eukaryotic gene expression is regulated during transcription and RNA processing, which take place in the nucleus as well as during translation, which takes place in the cytoplasm, and further regulation may occur through post-translational modifications of the proteins. Now, the main difference between the prokaryotic gene regulation and eukaryotic, that is lack of nucleus. Here, eukaryotic contains the nucleus. RNA transcription and protein translation occur almost simultaneously in case of prokaryote, while in case of eukaryote, RNA transcription occurs prior to the translation and it takes place in the nucleus. RNA translation to the protein occurs in the cytoplasm. RNA post-processing includes addition of 5' gap poly A tail and excision of introns and splicing of exons. Gene expression is regulated primarily at the transcriptional level in case of prokaryotes, while gene expression is regulated at many levels, for example, epigenetic, transcriptional, post-transcriptional, and translational and post-translational level. The figure shows the prokaryotes as well as eukaryotes gene expression and the presence of different uh, molecules in different parts of the cell. Prokaryotic gene expression is simpler, mainly controlled at the transcription level by operons and regulatory proteins, and occurs in the cytoplasm. In contrast, eukaryotic gene expression is complex and regulated at multiple stages. Epigenetic controls, transcriptional, RNA processing, mRNA transport, and translation occurring in the nucleus and the cytoplasm separately, allowing for the complex tissue and developmental specific gene control. Now, the details can be seen here. In case of prokaryotic gene regulation, they focus on transcription. For example, regulation primarily occurs by controlling the rate of transcription initiation through interactions with the activators and repressors. Operons, genes are often organized into operons, which are groups of the genes transcribed together and controlled by a single promoter and operator. Sim- while in case of eukaryotic gene regulation, multiple level control, regulation occurs at multiple stages, transcription, regulation of RNA polymerase binding to promoters, post-translational processing, splicing, and adding a gap and tail to mRNA in the nucleus, mRNA transport, movement of mRNA from the nucleus to the cytoplasm, translation, control over protein synthesis in the cytoplasm, post-translational modification changes to proteins after they are formed. While in case of prokaryotes, simultaneously gene transcription and translation takes place. Because there is no nucleus, transcription and translation can happen concurrently. Simplicity, prokaryotic gene regulation mechanisms are generally simpler, reflecting the fewer genes they process. Compartmentalization, the presence of a nucleus separates transcription from translation complexity. The eukaryotic cells require more complex and intricate regulatory mechanisms to control the large number of genes and enable diverse cell types and developmental processes. Then now it comes to the alternate splicing, RNA splicing concept, where exon and something like coding and non-coding region. For example, here you can see how it works. So exon 1, 2, 3, 4, and 5. Now, again, in DNA, in RNA, exon 1, 2, 3, 4, and 5. Then alternate splicing leads to 1, exon 2, 3, 4, and 5. Then another way of formation of after alternate splicing will be exon 1, 2, 4, and 5. Then another possibility is exon 1, 2, 3, and 5. And now there will be different forms of proteins formed. For example, protein A, B, and C. So the genes first were observed like that exhibited alternate RNA splicing. Alternative RNA splicing is a mechanism that allows different protein products to be formed or produced from one gene when different combinations of introns are removed from the transcript. This alternative splicing can be haphazard, or but more often it is controlled and acts as a mechanism of gene regulation with the frequency of different splicing alternatives controlled by the cell as a way to control the production of different protein products. Now, so there are some key terms which you need to understand. For example, alternate RNA splicing, codon, DNA ligase, polymerase, deoxyribose, double helix, epigenetics, exon, gene expression, genetic codes, helicase, intron, exons, a lagging strand, leading strand, mRNA, mismatch repair, mutation, nitrogenous bases, non-template strand, then nucleotide excision repair, Okazaki fragments, phosphate groups, post-translational modification, post-translational controls, primer, promoter, enhancer, RNA polymerase, rRNA, replicate forks, semi-conservative replication, splicing, start codon and stop codon, transfer RNA telomerase, then telomere, template strand, transcription, herbal. These are the details of the basics of molecular biology which you need to understand. And thank you.